بسم الله الرحمن الرحيم. Basic Structure of Cell. الدرس الثاني Lecture 2. هنا نرى here you can see the different types of organelles. Here are the lysosomes. Here we can observe ribosomes. Here is the nucleus. We can see here centriole. This is microfilament. This is the small plasmic reticulum. This is mitochondria. This is the rough plasmic reticulum. This is Golgi apparatus. Historically, cell theories were established by these scientists, Virchow, who specialized different type of cells. Nerve cell, this is a white blood cell, and this is muscle cell. In the year 1665, Robert Hooke used a microscope, to examine thin slice of cork. Cork means dead plant cells. These are dead plant cells which were examined using this microscope. What was seen looked like small boxes. Small boxes were observed, were seen by this microscope, made of cells. Here are made of cells, as well as all animals are made. In the year 1855, German medical doctor named Virchow observed under the microscope cells dividing. The division of cells were observed in the year 1855. All cells come from other pre-existing cells by cell division. So through cellular division, new generations of cells come from pre-existing living cells. All living things are made of cells. This is the cell that constitutes it's the basic structure, basic unit structure for all kinds of life. They are the unit structures and the units for functioning of all organisms. So it is a basic unit of life. Cells are the basic units of life. Cells come from reproduction of existing cells via cell division. These are simple cells, but here we can observe a complex organism constituted of many cells. Cells that lack nucleus or membrane-bound organelles are prokaryotes. They include bacteria, which are the simplest type of cell. They are single circular chromosome. Their chromosomes are characterized by that they are circular in shape and single. The nucleoid region center contains the DNA. DNA is contained in this center, which is called nucleoid. The nucleoid is surrounded by a peptidoglycan molecules that surround the nucleoid. Peptidoglycan molecules are made of peptides, which are proteins connected by connected with carbohydrates. Contain ribosomes. This nucleoid contain ribosomes. We can see no membrane here, so the nucleoid is stretched through all this area. No membrane, is not enclosed by any type of membrane. So ribosomes scattering here are used to make proteins. This is an eukaryotic cell characterized by a nucleus and membrane bound organelles. Include protists, fungi, plants, and animals. They are more complex type of cells. See, in prokaryotes are simple type of cells. Eukaryotic cell contains three basic structures. One, the nucleus. This organella is the nucleus. This is a cell membrane. As an envelope for the cell. Besides the cytoplasm, containing many types of cellular organelles. The main types of eukaryotic cells are plant cells and animal cell. This is an animal cell and this is a plant cell. Organelles are very small microscopic units performing different functions, different cellular functions. They are found in the cytoplasm. May or may not be membrane bound. Some types of organelles are membrane bound, but other types, or many types of organelles are not membrane bound. The cellular plasma is constituted of a double layer phospholipids besides proteins. Surrounds outside of all cells. All cells are surrounded by this cellular membrane. This membrane controls molecules and other types of matters that leave or enter the cell, leave the cell. So many types of molecules enter the cell from outside to inside, but other types of molecules in opposite direction are carried from inside the cell to the outside of the cell. As a living layer, this membrane is characterized as a living layer. This is phospholipid molecules composed of a fatty acid tail is attached to a glycerol head. Heads contain glycerol and phosphate and are hydrophilic, so they attract water. So instead of hydrophilic, we can say polar. This is a polar head that attracts water. Water is polar, and this is a polar head. And likes, these are likes, so water can be attracted to glycerol head. Cells are made of fatty acids and are hydrophilic. They repel water. Fatty acids are non-polar. Water is polar. Non-polar is polar are repelled. Makeup of bilayer where tails point inward toward each other. These tails are pointed inward here and here, here, facing each other. But the glycerol heads are located outside of the membrane and inside of the cellular membrane. Can move laterally to allow small molecules, carbon dioxide, oxygen, and water to enter the cell from outside to inside. Cell membrane is fluid. This is hydrophilic region of protein. This is hydrophobic region of protein. This area is mentioned as 5 nanometer long. And this is the phosphobilayer. These are the polar heads, and these are the non-polar tails. Proteins help move large molecules or aid in cellular recognition. These proteins help to communicate, to know different types of stimulators, recognize them, enabling cellular communication. And these proteins can be divided as peripheral protein and integral protein. Peripheral proteins are attached on the surface, and integral proteins are embedded completely through the membrane. Peripherals can be attached inner, outer, or inner in the cellular membrane. In plant cells, we can mention a cell membrane that lies immediately against the cell wall in plant cells. Pushes out against the cell wall to maintain cell shape. Cell shape maintenance is achieved through pushing out against the cell wall. Found outside of the cell membrane. The cell wall is found outside of the cell membrane. It's a non-living layer, but the cellular membrane is a living layer. The cell wall supports and protects cells, makes cellular support and cellular protection. It is found in plants, fungi, and bacteria. The cytoplasm here is a jelly-like substance enclosed by cell membrane. This jelly-like substance is enclosed or enveloped by the cellular membrane. provides a medium for chemical reactions to take place. All chemical biochemical reactions within the cell take place. Providing a medium by the cellular cytoplasm. The cytoplasm contains organelles, cellular organelles, that carry out specific cellular jobs. This is the cellular nucleus. Controls the normal activities of the cell. Contains the deoxyribonucleic acid, deoxynucleic acid, DNA, deoxyribonucleic acid, composed of ribose sugar and ribose and nitrogen base. We can mention these later. Components of this DNA, the monomers that constitute polymers of DNA. Chromosomes are found here. Nucleus. They are bound by a nuclear envelope. Nuclear membrane contains many pores. The nucleus is usually the large organelle. The largest organella of the cell is the nucleus. More on the nucleus. Each cell has fixed number of chromosomes that carry genes. Different animal and plant cells contain specific number of chromosomes. These chromosomes carry genes. Genes control cell characteristics. All cellular characteristics are controlled by the genes. The nuclear envelope is surrounding nucleus. This is the nucleus surrounded by the nuclear membrane, and these are the nucleus or the nuclear pores. Also called nuclear membrane or nuclear envelope, contains nuclear pores for materials to enter and leave the nucleus. Many materials enter the nucleus from outside to inside the pores. Other materials go out from inside to the outside of the nucleus through these pores. Inside the nucleus, you can observe the chromatin, which can be organized into chromosomes through the phase of cellular division. DNA is spread out and appears as chromatin in non-dividing cells, but in dividing cells we can mention these chromosomes. This is an image of DNA molecules, which is seen here as a double helix. Here, this is one helix, and this is another helix, which go on opposite directions. They are antiparallel, constituted of pairs of nitrogen bases attached by hydrogen bonds through a backbone of sugar phosphate. DNA is the hereditary material of the cell. Genes that make up the DNA molecule code for different proteins. Different proteins are encoded by the genes. Here we are observing the nucleus. It is composed of pores found within the nuclear envelope, and this is the nucleus, and this is chromatin scattered or found within the inside of the nucleus. The nucleolus is found inside the nucleus and disappears when cell divides through the phase of cellular division. The nucleolus disappears. Makes ribosomes that make proteins. Ribosomes for making proteins are made by this nucleolus. These are the microtubules and these are the microfilaments. All these together constitute the cellular skeleton. Here you can see the centrioles found only in animal cells, but plant cells contain no centrioles. They are paired structures near the nucleus. Found near the nucleus, you can see these organelles. They are made of bundle of microtubules. Microtubules are attached beside each other, constituting centrioles. They appear during cell division, forming mitotic spindle. The mitotic spindle through the cellular division are made by these organelles. They help to pull chromosome pairs apart of opposite ends of the cell. Chromosomes are pulled to each pole of the cell by these centrioles, enabling division of living cells. Eukaryotic living cells. The mitochondrion, single, but mitochondria is plural, is organella. They are the powerhouse of the cell. All the energetic currency of the cell are made by this organella. It's called the powerhouse. It generates cellular energy in form of this molecule, this compound, which is named as adenosine triphosphate. More active cells like muscle cells have more mitochondria. Mitochondria are found within active cells like muscle cells. Both plants and animal cells have mitochondria. Site of cellular respiration, or the burning of the fuel, burning of glucose. Glucose burning is performed here in this cellular organelle. The mitochondria is composed of an outer membrane, outer mitochondrial membrane, and an inner membrane. Here are cristae. These are many cristae, and inside the mitochondria is called the mitochondrial matrix. Mitochondria is surrounded by a double membrane, and it has its own DNA, which is circular DNA, mitochondrial circular DNA. It's folded inner membrane called cristae that increases the surface area for more chemical reactions. More chemical reactions can occur through increased surface area performed by these cristae. Mitochondria come from cytoplasm in the EGG cell during fertilization. When spermatozoon enters an ovum, when entered by spermatozoon and fertilization occurs, mitochondria come from cytoplasm in the EGG cell. Therefore, you inherit your mitochondria from your parents. Your parents inherit your mitochondria. The endoplasmic reticulum, it is a network of hollow membrane tubules, connects to nuclear envelope and cell membrane. Cell membrane and the nuclear envelope are connected by this reticulum, the plasmic reticulum. This reticulum is functioning in cellular products synthesis and transport. The rough endoplasmic reticulum is distinguished by scattering ribosomes. When compared to the smooth endoplasmic reticulum, that contain no ribosomes. These ribosomes make membrane proteins and proteins for export out of the cell. Rough endoplasmic reticulum is found attached to the nuclear envelope of the ribosomes. It's the rough endoplasmic reticulum. It's a smooth that contain no ribosomes, but the rough endoplasmic reticulum is characterized by scattered numerous numbers of ribosomes. Proteins are made by ribosomes on the endoplasmic reticulum surface. They are then threaded into the interior of the rough endoplasmic reticulum to be modified and transported. These proteins are modified and transported by this endoplasmic reticulum. Functions of the smooth endoplasmic reticulum makes membrane lipids, such as steroids, and regulates calcium muscle cells. In muscle cells, calcium is regulated by this organelle. Toxic substances are destroyed in the liver also by this organelle. Here we can see the endomembrane system. This is the nuclear envelope. This is the nucleus. These are ribosomes. And this is smooth endoplasmic reticulum. This includes nuclear membrane connected to endoplasmic reticulum, connected to cell membrane transport. Ribosomes made of proteins and RNA, proteins and ribosomal ribonucleic acids. Protein factories for cell. The machinery for protein synthesis are the ribosomes. Through joining amino acids, constituting or making proteins via protein synthesis. Ribosomes can be attached to rough endoplasmic reticulum. You can see here more bright image, or be free and attached in the cytoplasm, scattered within the cytoplasm. Also, we can mention these ribosomes scattered in the cytoplasm of the cell. Golgi bodies, this is Golgi apparatus, this is trans, and this is the same direction. Sacs having shipping side, cis-face, and receiving side out face. This is receiving side, and this is shipping side inside base of Golgi apparatus. They receive proteins made by endoplasmic reticulum and transport vesicles with modified proteins bunch off the ends using transport vesicles. Package store sorting and packaging of the molecules from the plasma reticulum for storage or transport out of the cell is performed by Golgi bodies. The lysosomes contain digestive enzymes. This is a lysosome. It breaks down food, bacteria, and worn out cell parts for cells. Also, unneeded waste is going to be thrown out using these lysosomes. And programmed for cell death, the lyse release enzymes to break down and recycle cell parts. Recycling of cellular parts is performed by lysosomes. Lysosome digestion. Cell take in food by phagocytosis. Food can be entered inside the cell through phagocytosis. Lysosomes digest the food and get rid of waste through exocytosis. Cilium flagella. Cilium flagella can function in moving cells, in moving fluids, or in small particles across the cell surface. Small particles across the cell surface. The cell surface are moved in function in moving cells. The cellular movement cellular movement is performed by these organelles. Cilia and flagella. Cilia are shorter and more numerous on cells, but flagella are longer and fewer in number, usually 1 to 3 on each cell. So moving cilia and flagella is combination moving cell is moving spermatozoa. Cilia moving away dust particles from the lungs. This lung cell, and this is the mucous gland. And these are cilia pushing away dirt from the lung out to the mouth. Vacuole. Here is a large vacuole. Fluid-filled sacs for storage different cellular materials. They are small or absent in animal cells. Plant cells have a large central vacuole. This is a large central vacuole found within a plant cell, but in animal cells we can find no vacuole or small vacuoles. These are vacuoles. Just like something container cellular matters. Plants store cells include storage of sugars, proteins, minerals, lipids, weights, salts, water, and enzymes. This is the chloroplast here. Found only in producers, organisms containing chlorophyll, producers of the carbohydrate. Just as green plants, all green plants contain this chlorophyll. Chlorophyll use energy from sunlight to make glucose. Energy from sun stored in the chemical bonds of sugars. So through breaking chemical bonds of sugars, energy is achieved by animal cells. Chloroplast surrounded by a double membrane, and the outer membrane is smooth. This is smooth outer membrane. The inner membrane modified into sacs called thylakoids. The inner membrane is organized as thylakoids, which are sacs called grana, interconnected and interconnected. Stroma, gel-like material surrounding thylakoids.